Okay, so you have already followed my instructions and you have cut a piece of paper down the middle. So I should have 2 long pieces of paper now. Now you're going to stack those 2 pieces of paper on top of one another, just like this. And then you're going to take that very long strip of paper and you're going to fold it in half. Once you've done that, unfold it. You're going to take this top portion and you're going to find that crease and you will stop at the crease. Make sure it's straight and put another crease in. Unfold it. Same thing on the other side. You're going to take this long piece, you're going to fold it into the middle crease. Line it up, stop it, and put another crease. So when you're done, you should have 1, 2, 3, 4 sections. I'm going to do this twice. I want you to just watch me this time. So what I'm going to do, and you're just watching, you're not participating, you're going to slide this other sheet and you're just going to slide it off. So now I have 2 identical sheets. I'm going to take this one and I'm going to pull it down, pull it down, pull it down, and I'm going to flip it over just like this. So now do that with me. So I have both my sheets. I'm going to slide the one off of the other, and then I'm going to pull the one down, 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 and then when the bottom of this one and the top of this one meet, I'm going to flip one of them over on top of each other, just like this. Go ahead and do that. I'm out of glue, so what I'm going to do is just put a little piece of tape under mine for right now, but you'll put a glue on there and then stick it back like it's supposed to be, meeting into the crease, because you're going to be folding this, meeting into the crease. So now you have an extremely long piece of paper with a bunch of folds. So with me, we're going to make a fan. We're going to fold it forwards, backwards, forwards, backwards, forward, backward. So now you should have this. Let's do it one more time. Fold it forward, backward, forward, backwards, forwards, and then backwards. So now you have this fan. Remember the frog legs in preschool? Well, you just made frog legs. That's all you're doing. Okay? Super easy. This is what you should have. If you don't have something that accordions like this, you need to talk to the people at your desk and fix it. So our ultimate goal is to make the cell cycle. And this is one that a student did for me. So this is your ultimate goal right here. So what we're showing is, on the top panel, we're showing the entire cell cycle and what it goes through. And then we're showing the first stage, the next stage, the next stage, the next stage, the next stage, and until we finally get 2 daughter cells here in the bottom. And all along, the student is telling me what's happening. And they're also noting with the title, each one of these squares, if I look at it just as a square, each one of these has their own title. So let's do one together and I'll show you what it looks like. Okay, so I'm going to skip a window because this is my entire cell cycle, and I want to go and just concentrate on for a second interphase. So I'm going to follow my directions exactly how it says on this sheet. So first I'm going to have my title. I'm going to use my handy ruler because I know I need it. I am not neat. Okay, so the title for this is Stage 1, interphase. Okay, I'm going to go and fix the things that I couldn't do, like my P, make my tail bigger, my G, make my tail bigger. So stage 1 interface, and I need to write a little bit about that. So I'm all for being really neat. So this is 11 centimeters wide, so I'm going to take 5.5 centimeters right here, here, and I'm actually going to do this just because it helps me. Do a thin line, and I'm going to come over here and I'm going to write everything that's going on in stage 1 interface. So during interphase, cells grow into a mature, this is why we use a pencil, size. It also makes a copy of DNA and prepares to divide into 2 cells. So I have everything written to the side. Again, pencils are great. So now I'm going to come through and I'm going to draw a cell. So you might have something available to you. I don't have something available for everyone. So just do the best you can. There's my cell. I'm going to draw my nucleus. My nucleus is quite big. Why? Made a copy of it. So here's all the little strands of all my DNA. I have centrioles. And spindle fibers here, growing and beginning to move. So I need to draw that because my book talked about it. So as you can see, of my main structures for this drawing, I have my spindle fibers, my centrioles, and my nucleus, and it looks nice and neat. This is what it should look like the entire time I am looking at your drawings. Nice and neat. Some people choose to put their cell in the middle and then they write around it. That's fine as long as I can tell a difference between this and this, and you find a way to separate that. Everything will be found in your book as well as your notes, and if you still can't find it, to the Google you shall go. So your first window is the cell cycle. Your second window is stage 1 interphase. Then underneath that, so each of those is a window. So cell cycle is your title, stage 1 interphase. The next title, stage 2A mitosis, which is known as prophase. Stage 2B mitosis, known as metaphase. Stage 2C, mitosis, anaphase. Stage 2D, mitosis, telophase. All 4 of these, write them exactly how you see them. Every year I'm asked this, and every year I say the same exact thing. This part here where it says no pencil, cross that out. Why wouldn't we use a pencil? Use the pencil. Because as you can see, I had to erase quite a bit. So these all need to be named exactly how you see them. These are your titles. On the back of your paper, you get 4 points for a title, name, date, and period. That means the hour. For this second one, you should have stage 1 interface, title, illustration, colored and labeled with the proper description. And each one of these is the exact same thing. Next, we have follow directions. Were the lawns drawn neat and were they straight? Was it colored neatly? Did you fold it neatly? Folding neatly is something that you should all be able to do in high school. Did you have your grading rubric? That means if I had to give you a second one, I'm going to take a point off for not being responsible. Did you write legible? And as far as this is concerned, this is a bonus, just for being here. I think you will find the cell cycle to be extremely frustrating because you really have to think where you want the placement of everything to be. You really have to look at your book and your notes and perhaps even go back to some videos and take a look-see of how exactly does that stage work again. What organelles are involved in that stage process. And I think that's probably the most frustrating things for students. This project is really simple to do, but it is time consuming and it can be very frustrating. So I do recognize that fact. I will tell you on the end game, this will really help you with your test, and your EOC, unlike anything else I could possibly give you. So that's why I've continued to do it year after year. If you have any questions, this is an in-class project. I'm right here.